thank you very much to the organizers for inviting me, and to all of you for listening. So I'm a plant geneticist, and like John, I find this plant really exciting, and it's interesting to me to come here. I think this is a great group of people with different interests, different skills, different backgrounds, and some of you in the audience are really helping to build a solid foundation of our understanding of how this plant affects us, starting with one enzyme. Starting with one cannabinoid, or maybe one terpene at a time, and understanding how that works with our own CB1 and CB2 receptors and other aspects of our physiology to create an effect. Other people are starting with the plant side of things and taking the whole messiness and complexity that's in the plant material, or parts of that, and seeing how that affects people and using that as a treatment. And I think before I really get into the main message that I want to say, I think it's really important to think about things starting from both directions. I think we know that the plant has a lot of important effects on a lot of different key traits, key pathways in our own bodies, but we don't really understand how it works because of the complexity of all of the different terpenes and cannabinoids that we just heard about, actually. And on the other side, we know that the effects of some key molecules, but if it was as simple as one molecule creating a lot of these effects, marinol would be success. And almost nobody uses marinol, right? Because it doesn't have some of the same properties of the plant material, and we don't really understand why. So keep that in mind as I'm talking. So just thinking about how I got here, when I started as a professor a few years ago in Colorado, a lot of changes were happening on the legal side, and just as we all know, a lot of different states are starting to experiment with this and creating these state legal markets where we can start to see what people are going to basically, we're starting to see the effects of some of these state markets, and people are starting to do some science. But the science that's being done now really dates back a lot. Cannabis science there's mentions of cannabis in the oldest medical texts in both Asia and Europe. So it's something that's been recognized for a long time, that there's something important about this plant, and yet we still don't really understand very much how that's working. And that's despite really a lot of great work by people, but it's a complicated system. And today, in the US anyway, most of the science that's being done on the real clinical side of things has to go through the National Institute on Drug Abuse, which is the sole legal provider of most kinds of cannabis products for medical research in the US. So this is 1968. This one source in Mississippi has been really where all marijuana products for most kinds of studies on human physiological or animal effects are coming from really this one source. And most published studies used a very limited pool of cannabis diversity with only 2 or 3% THC, anyway, very low levels of THC, and also very low levels of any other cannabinoid. Also, very anecdotally anyway, very little in the way of terpenes or other key plant compounds. NIDA has recognized finally that this is maybe an issue, and they've been trying to increase some of the THC levels and other cannabinoid levels and diversity. However, I think it's what I wanted to do as part of this is to think about how relevant, or how how what's available from NIDA compares to what's available in the state markets, and how relevant is that, or when we try to compare what's in these medical studies to what people are saying anecdotally, how do we make that connection? Just to put things in perspective, I thought I'd look at how many different papers talked about medical health effects, and the National Institute on Drug Abuse derived some of their findings either directly or indirectly from information derived from these strains. And we're talking about thousands of papers every year are at least mentioning marijuana health effects and National Institute on Drug Abuse. It's not necessarily definitive, but I'm just pointing out a lot of people are using information derived from this to make some kinds of claims, scientific claims. And mostly this is focused on, mostly it's THC and now CBD, a little bit of work on CBG and CBN, but these are basically the decarboxylated breakdown products of some of the and they're more typically thought to be more bioactive than the things that are in the plant, but basically a limited number of cannabinoids people are talking about. And we know something about the genetics of, if you have a hemp strain, you might have some variation in how much CBD is there, but typically most of the cannabinoids are going to be CBD, very little THC, whereas marijuana strains are going to be mostly THC, very little CBD, and if you cross them, you get a whole mixture of different genetics. So each of these dots is a different genetic variety that in these cases have novel cannabinoid profiles different than either of their parents, and that's really the basis of a lot of modern cannabis breeding. But really, most of the work that's being done is only looking at a very limited part of the diversity of cannabinoids. So this is just looking at tiny. These are all different cannabinoids that you'd find in the plant. These are just a small section of the diversity, and this is their biosynthesis. And then these are some different breakdown products that can happen. And you can see these are some different terpenes, and you can see that they're derived from some of the same precursors, so related biosynthetic pathways. But this is just a tiny portion of the chemical diversity that's in the plants that's really being looked at. But there's a reason for that, of course, is that these are some of the major cannabinoids in there. I just want to mention some great work that Ryan Lynch did when he was in my lab. Now he's at Medicinal Genomics doing great work there. But basically what I want you to get out of this complicated slide is that there's some genetic lineages. This is based on genome sequencing of a large proportion of the diversity. Of course, there's always more that you could wish for to be included, but we were able to pull out hemp lineages as being distinct from broadleaf drug types and narrowleaf drug types, the things that have a lot of THC in them, and then a lot of things that are hybrids, mixtures of different genetic components. So this is called a genetic network, showing some of the connections representing the major genetic lineages. What really I want to get at is that there are some. In this case, we were able to identify 3 different genetic lineages that were pretty clear. And if we look at the chemical diversity, we can see they differ in CBDA and THCA, right? So there's some differences between broadleaf and narrowleaf, but also bigger differences between either of them and hemp in terms of how much THC. Of course, hemp has very low THC and much higher CBD. This is just a limited sample of diversity, but still I think tells somewhat of a clear story. And if we look at the more minor cannabinoids, we can see that those same 3 lineages differ in some of, so the precursor molecules just basically vary a lot. How much is in the plant material is going to basically depend more on when you harvest it and how you grow it, rather than any genetic differences. Some of these different minor cannabinoids actually do at least in our sample, differed between the major genetic lineages, and others didn't. That's basically where I'm going to leave it at that. Is that there could be some key diversity differences in some of the more minor cannabinoids. Similarly with some of the terpenes, again, there's some things that vary a lot within the different lineages and are not significant, so the same letter means they're not significantly different. The biggest difference, at least in our sample, in the terpenes was in myrcene, and several others have suggested that might cause some of the differences between some of the major genetic lineages in terms of some of their effects. Could be due to myrcene, but who knows? And I want to keep that in mind, that these terpenes and terpenoids, they're mainly responsible for the odor and flavor, but they have antimicrobial properties. Some of them are already known to interact with the CB1 or CB2 receptors, and also do seem to interact with the cannabinoids, based on some limited evidence, to lead to alterations in the psychological effect. And the science behind that is pretty limited. There's some, but it's limited. But the main thing is, if you go into a dispensary, you'll find a lot of different strains that have very similar levels of THC and yet have dramatic differences in their effects. And so it's not just about THC. It's about something related to some of these other compounds in there. Very well might be some of these terpenes, as it turns out. But I wanted to compare, okay, so there's all this diversity out there. What kind of diversity is available for federally legal research at universities? And how does NIDA compare to what's in the state markets? And just to show a few of the cannabinoids, if we look at CBD, and we had to calculate CBD levels because the federal government only releases CBD. They don't break it down into CBD versus CBDA or THC versus THCA because they use GC technology to measure their cannabinoids. And so what they report doesn't include that information. So we had to convert our values to equivalent decarboxylated versions. Just for those of you who care about that, I know there should be an A after it, but in this case, the data we have is on CBD from the federal government. And if we compare what's available, you can see they actually do a pretty good job for CBD in terms of the range of different values. So each of these represents a different NIDA strain and their reported levels of CBD. And you can see there's a fair amount of diversity. On average, their CBD lines are lower in CBD than what you get in state markets, but at least the range of diversity is there. If you look at THC levels, you can see it's much lower than anything that would ever be sold by a reputable dispensary anymore. I mean, it's about a quarter as much as you'd ever get in a dispensary in most of the cases, except for, of course, it's more similar to the THC levels in like 1-to-1 or variable lines that have a mixture of THC and CBD in some of these dispensaries. But on average, 5 to 10% is on the high end for what NIDA makes available for research. CBG is not that different between the federal and the federal materials and the state markets for at least the lines that have CBG, but they don't have anything that they've listed that has substantial THCV, which is potentially a quite interesting other cannabinoid, but not present in any of the NIDA material according to their results. And so that's sort of the averages. What about another way of looking at the diversity is for lines that have both CBD and THC in them at more than 1%. So taking out the lines that are just pure CBD or mainly CBD or mainly THC, what kind of diversity does NIDA offer of things that have more than that? And you can see it's only low levels of total cannabinoids and also only a subset of the range of variation in CBD to THC ratio is available in the lines that they have listed compared to the broader diversity available in the state markets. So it's not necessarily totally irrelevant, but it only tells us a part of the story of why people are using cannabis products in the state markets, and also the potential range of effects of those cannabis products, both good and bad, is not able to be studied using this kind of material. If we go broader and look at all of the total cannabinoids, it's basically the same kind of story. So basically, this is taking a whole broad range of cannabinoids, distilling it statistically down into only two axes, principal component 1, principal component 2. And what that does is this just tells you, it helps us visualize the diversity of the phytochemical diversity available in some of these different markets. So just comparing Oakland, Sacramento, and NIDA, we can see that NIDA, again, only has a tiny range of variation compared to what's available in the state markets. And these are, and you can see also a lot of the state markets have also these outliers that are even more unusual plant material, whereas the NIDA material has very little in the way of these extreme outliers, and really only covers a very small range of the total diversity, the phytochemical diversity that's present in the state markets. So just to think about that a little bit more, we've heard today about the effects of individual cannabinoids or groupings of cannabinoids and terpenes, and some of that is certainly done at the university level, but a lot of what we hear about is, I guess a lot of what you read about in the papers is going to be limited because NIDA has very different levels of cannabinoids. They have roughly a quarter of the THC levels in their THC lines. They typically have lower CBD levels than what you get at a state market, but I would argue that it's not as extreme as the THC situation. Essentially, no measurable THCV, CBC, or other what some people term minor cannabinoids. I should also point out that they don't even report terpene levels for their material that they send out, and so you can't request material easily, at least, to have a particular terpene content. I think the research using NIDA marijuana is limited in terms of what kind of terpene diversity is there. There are some things that I haven't shown you from NIDA. There are some cannabinoids that are actually higher in NIDA. So actually one in particular, NIDA has 11 to 23 times the CBN content. So what does that tell you? Yeah, it's old. Oh, bold. Well, it's also old. So CBN is a natural breakdown product of delta-9 THC due to oxidation or UV. Other processes occur over time, leading to CBN to break down. So one possible explanation, using NIDA's information from NIDA's website that I got just a few days ago, they say the university designates a secure plot of land where marijuana crops are grown every few years based on current and expected demand. The marijuana is grown, harvested, stored, and made available as bulk marijuana or other purified elements of marijuana to use for research. Every few years. Okay. Maybe that explains why it has 11 to 23 times as much of the breakdown products of cannabinoids as anything you'd ever see in a dispensary. And this is not I would say this is not necessarily an innocuous issue, because we know all of these cannabinoids are interacting with each other to create the physiological effects. And the fact that the research grade materials are so heavily degraded that they don't have substantial terpenes and have much higher rates of the breakdown products limits our ability to really understand what's happening when people are using very different material in the state market. So I would argue that this issue, NIDA having lower levels of most cannabinoids, a lower diversity of cannabinoids, and higher levels of the breakdown products of THC and CBD suggests, I would argue that's a major issue when we're trying to understand the effects of the marijuana that people are using in the state markets. And I would advocate that something needs to change such that ongoing and future research can be improved in terms of its relevance. We need materials with higher diversity of cannabinoids, higher levels of relevant products, lower levels of degradation products, unless you're interested specifically in studying those degradation products. And also making sure that we have relevant levels of the key terpenes and terpenoid compounds. Just to briefly thank people before my time totally runs out. Daniela Vergara, my postdoc, did the bulk of the statistical analysis, along with Cinnamon Bidwell, and my collaborator in the Psych and Neuroscience department at CU, Kent Hutchinson. Also, the genetic analysis that I mentioned at the beginning was done by Ryan Lynch, and funding from several different sources, and although mainly actually from donations. And also, I really should thank Steve Hill for their expertise on the chemical side of things and their help with understanding and generating a lot of this data. So thank you very much.